Welcome to a very special lecture, the Marshall W. Nirenberg Lecture, here in Missouri Auditorium on the campus of the National Institutes of Health. And hello also to all the people who are watching on the video. Wonderful to see Missouri Auditorium absolutely packed with people here this afternoon, which I think reflects the importance of the topic and the speaker that we're fortunate to have with us, namely George Church. Before I introduce George, let me just also say a word about Marshall Nirenberg. I'm one of those who had the good fortune to know Marshall, and of course, he is the individual who figured out the genetic code back in the early 60s, beginning with that realization of that poly-U codes for phenylalanine, and going on from that to fill out the table, something that we have had many occasions to reflect on since Marshall is one of our own, having spent almost his entire scientific career here. And if you've not seen the display about him and his work that's located over near the Lipset Auditorium, in case you've not wandered over there, go have a look. It's pretty interesting to see some of the original materials and equipment that he used back then, more than 50 years ago, to figure out this fundamental information about how information gets translated from nucleic acids to protein. And Marshall, as you know, did a lot of that work in the area of molecular biology, and in the second half of his career became fascinated with neurobiology, and was one of those individuals with just boundless curiosity. And I certainly remember a number of conversations with him as he was not only chasing after the secrets of neurobiology, but bringing into that a whole new set of ideas about chemistry and small molecule approaches, which were a great deal of fun to talk about. So we've had, since the last 5 years, this lecture to honor him in particular, and we have tried to make this an opportunity to bring in some of the most highly respected figures in the fields of molecular genetics. And today we have actually done extremely well in that regard, because we have a speaker who's going to speak about genetic codes, but also about brain codes, so capturing neatly both parts of Marshall's career in the work of Dr. Church. He got his undergraduate degree at Duke. He went on to get a Ph.D. at Harvard in biochemistry and molecular biology. After a brief stint at Biogen and then at UCSF, he landed back at Harvard, where he has been since 1986, and is now professor of genetics, health sciences, and technology. He's director of the Harvard NHGRI Center of Excellence in Genomic Science, having had that role now for all of 13 years. Well, this is a major investment the Genome Institute has made into development of technologies, and George has had a wonderful team that's been working on this, and no doubt we'll hear something about that. He's director of the Personal Genome Project, the first effort to derive complete sequences on individuals and put all that information truly in the public domain, including his own. He's a senior associate at the Broad Institute, and he is also founding core faculty and platform lead in synthetic biology at the Wyss Institute for Biologically Inspired Engineering. I am happy to say I think I've known George for 30 years, and the way in which we, in those days, were trying to figure out how to sequence DNA at the proper speed that it might ultimately lead to the whole human genome. And as you know, that ultimately succeeded, and in no small part because of the creativity of George and others who continually invented new ways to speed up the possibility of getting rapid and accurate DNA sequence. In fact, he developed one of the first direct genomic sequencing methods. He's elected member of the National Academy of Sciences and also the National Academy of Engineering. And he was cited as one of the major contributors, which I suspect he may say something about, in Science's Breakthrough of the Year, namely CRISPR, where George has played a very significant role in the revolution that probably almost everybody in this room is now using in their own research. He has no less than 70 patents. So we are fortunate indeed to have Dr. Church with us. Please join with me in giving him a warm NIH welcome.
Thank you very much, Francis, and everybody. It's nice to see old friends here. You've already seen on the pre-slides my conflict of interest. That's what this is as well. There's actually a full web page dedicated to it if you want to look at that. And there will be some drugs mentioned, although it is by no means the point of the talk. It turns out that Marshall Nirenberg and I have had some pretty close related passions. I've been working on genetic code since I was a teenager, essentially my entire career, mostly initially on the crystallography of the first folded nucleic acid, which is transfer RNA, which is the thing that breaks this code. And so we'll start on genetic code and how far it's come and where it's going. And then he had many papers on neuroblastomas and in particular on transcription factors in neurobiology. And that's, again, our approach is transcription factors in neurobiology. And then finally, he had an interest in science and society, and I'll mention that briefly in wrapping up. So this is the genetic code 4 years after his first codon was dissected, which was UUU for phenylalanine. And you can see there's a lot of ambiguity here. There's a lot of things they're not sure whether it's nonsense or not, especially the leucine, tryptophan, serine, and arginine codons. But most of it was done by actually synthesizing each of these trinucleotides. So synthesizing 3 nucleotides was a big deal in 1965, and they made essentially all of them. And Khurana even made polymers of these trinucleotides. This was, and by the way, Phil Leader was one of the co-conspirators in this effort, and he was my first department chair. And so we're constantly reminded of the genetic code. Now this is where it is today, much less ambiguity. In fact, we have now the numbers here are the number of instances of each of these codons in a complete genome. And we have these numbers for many different genomes. This happens to be E. coli. And the blue boxes represent some of those codons that are ambiguous in the previous slide. And now they're the ones that we're targeting for removal, coincidentally. So they're leucine, serine, arginine, and stop codons. We have now synthesized the entire 4 million base pairs, for which these numbers represent the number of codons used for each type of amino acid. And in so doing, we've either eliminated 1 codon genome-wide or 7 codons genome-wide. And I'll give you the reasons in a moment why we did this, but the thing that's significant here is we're not only reading essentially the codons, but also now writing them. And we can do this radical recoding for reasons that we'll get to in a moment. And the reason that we can do this, the reason that we can read whole genomes and, in fact, write them sort of at the billions of base pair scale, is that this exponential. And this exponential was breathtaking even back in the 1980s. We didn't fully recognize it was going at Moore's law rate. And then around 2003 and 2004, it changed slope, which tells you to be suspicious of these straight lines. So this is factors of 10 on the y-axis. And now it's been about a 3 million-fold improvement in sequencing and about a billion-fold improvement in synthesis of oligonucleotides. And so I'll speculate on what happened in 2003 to 2004 in an extremely immodest way by focusing on papers that came from my lab in 2003, 2004. But it also indicate, and the key was going to chips, essentially perfectly flat surfaces, which allowed us to miniaturize, multiplex, and start a new Moore's law curve that was much faster. And this was, we were using chips to synthesize DNA, not to use them the way they were conventionally used, but to strip off the DNA so it lost its structure so that we could assemble large genomes. And there's an interplay between sequencing and synthesis. In fact, the next-gen sequencing is called sequencing by synthesis. Now, if we go from synthesizing short pieces to putting together large genomes, like the one that I briefly mentioned, this is often called editing. I think there's a version of this which is called writing, where you edit so much that you really have to synthesize huge blocks of 50, 100 kilobase is a megabase. But the editors, there's a lot of obsession recently, and Francis alluded to this with CRISPR, which is in the upper right here. But I want to remind everybody that there are at least 9 editors, and there probably will be more. And each one of them has been displaced or found its niche. And they have 3 different ways of scanning the genome, the 6 billion base pairs in the case of the human genome, to find a unique place to make an edit or a cut. And that scanning head, essentially the information interface, is either DNA, RNA, or protein. Examples of protein scanners are zinc finger nucleases, and RNA is the CRISPR-Cas9, and DNA scanning are things like the beta recombinase that I'll show you in a moment. Now, there are problems with CRISPR, and I will, even though my lab was among the labs that invented it, I'll be the first to point out the problems of all of our inventions. And this one has off-target issues and on-target issues. The off-target issues, there have been many solutions. We won't go through them. But all of them claim to be, or many of them claim to be so low that you can't detect the off-target. But that's typically because they've used the trick that was used in the very beginning here, 2013, which is computational, avoiding being off by more than 3. So you have no instances where you have 1 or 2 mismatches. There is now one way, if you're interested, of a general way of getting single nucleotide polymorphism, single nucleotide changes, specificity, and that's published in this top-rated journal called bioRxiv, which I encourage you all to publish in from my group. So that's single nucleotide specific CRISPR. And then there is the on-target problems, and this comes from all the methods that use a double-strand break, the zinc fingers, meganucleases, talons, and CRISPR. When you make a double-strand break, it's a race between the cell fixing it and getting your donor DNA to fix it how you want. And there are two alternatives that we've been working on longer than we've been working on CRISPR, and these are, as many things, these are harvested from the microbial world, in particular E. coli lambda beta recombinase and lambda integrase, and they work in different manners to avoid the double-strand break dilemma. And so the various integrases require pairing of donor editing molecule and the genome that you're trying to engineer. And the beta recombinase works at the lagging strand to pretend to be a Okazaki fragment. And so you're not making a break here, you're just incorporating a mismatch. Or it could be a very large mismatch. So we've actually used those 2 non-CRISPR methods, and so I call this beyond CRISPR, to do the largest and most radical genome engineering to date, which is changing the genetic code, back to Marshall Nirenberg, the genetic code of E. coli, and hopefully many other organisms soon, and the reasons are given here. And we've changed it first to get rid of 1 codon genome-wide. So there's 64 triplet codons, and all organisms share one thing. They don't all share exactly the same genetic code, although it's very similar, but they share that they use all 64 codons somehow. And in nature and synthetically prior to this work, there was none that used 63 or less. So now there's one that uses 63, and it shows all the things, all the goals that we had. It can use non-standard amino acids, not limited to the original set that all organisms have, 20, 21. It is genetically and metabolically isolated, so this can be used for biocontainment. And finally has multivirus resistant. I think this is very interesting both practically and philosophically, that you can make an organism resistant to all viruses in the world, even viruses you've never studied before, because they all expect a genetic code to be provided by the host. And we can change that, and the more radically we change it, the more viruses. But we were surprised that even changing 1 codon was enough to make it resistant to most classes of viruses. Now, you might say, well, why do we need new amino acids? I mean, isn't the 20 adequate? So I'm going to just quickly justify that, one of the 4 goals. These are some of the ones that we and others have used to advantage. You can put in fluorescent amino acids, which means, and you can put in multiple fluorescent amino acids, so it's actually more fluorescent than GFP and faster. Redox, there's a tenfold improvement in redox in some cases. You can replace disulfides with disolanides, which means that they actually fold and link in the cytoplasm, which is very reducing. You can get optical wavelength isomerization. You can get peptide nucleic acids, but in an inexpensive route. And finally, you can get these conjugates, and where you can make click chemistry or this ketone hydrazide coupling, and this is already in use in the pharmaceutical industry to make pegylated human proteins, like this case, human growth hormone. Now, in order to do a whole genome, the synthesis isn't so bad. It's bad, but it's not so bad, and it's getting better exponentially. The real trick is debugging the biology. So we wanted to de-risk this combinatorially. Rather than build a whole genome that was broken in 100 different ways and then trying to figure out what's wrong with it, we would break it up into pieces and debug all the pieces. And interestingly, when we did the one codon, it was everything, essentially all 320 changes worked. So that was a case where we were pleasantly surprised. When we did the two, what we thought would be the two hardest codons, they were indeed hard. We changed these are UAG is the rarest codon, and the AGG and AGA are the second and third rarest ones. They're arginine codons, not stop. And there were 13 examples where those were problematic, and we had to debug it, and eventually came up with workarounds for all of them. Then we did 13 codons for a huge fraction of the ribosomal genes, and here we had only one gotcha in changing those 30, and we engineered around that. So all of these things we engineered around, and that allowed us to move on to the whole genome, which is now 100% synthesized and assembled in yeast into 50 kb fragments. It's 86%, these numbers are a little out of date, but they're close enough, 86% in E. coli, and then we validate by incorporating them into the genome. And this was a team effort led mostly by Nili Ostrov, who's a postdoctoral fellow in the lab, as well as Michael Landon. Now, our goal is not just to have some wimpy genome that barely is alive. We actually want it to be faster than the laboratory strains, and here you can see is our inspiration for this. On the left-hand side, you'll find an organism that's disturbingly faster than E. coli, which is disturbingly faster than every other organism on the planet. This is called Vibrio nitrogens, and it's now becoming the workhorse, for molecular biology that may replace E. coli, and we will be recoding its genome or making E. coli have the properties that we want. So, to the second topic, having to do with Marshall Nirenberg, was brain codes. And I capitalized the word brain. For some of you may know, a few of my colleagues and I had an obsession with brain activity mapping around 2011, which managed to make it to the attention of the OSTP and NIH and various other agencies, and it became a real thing, which is now called Brain Research Through Advancing Innovative Neurotechnologies. So that's the acronym, and I really loved it when it was announced, because it included the words innovative and technologies, which is not something you often get from a top-down project. And it's shaped up to be very, very remarkable project, and I'll tell you a little bit about our little piece of it. So Marshall Nirenberg worked on these transcription factors, homeodomain factors, Krebs, and so forth, and I'll tell you what we're doing now and in the near future that specifically involves building brains, not just analyzing, but both building and analyzing. So it's the whole reading and writing mantra that we had for the genetic code and the genome, we can also do for this epigenetic code. And as you'll see in a moment, we tried various ways of doing the epigenetic reprogramming, including using CRISPR, but we found that in direct side-by-side comparisons, we could get a lot more mileage right now using transcription factors. And we were shocked to find out that despite the fact that we've got cDNA sequences from for many human cDNAs and nearly complete human genome sequence, there was no collection of human genes or human transcription factors that was adequate, even close to adequate. So Alex, as a graduate student in the lab, put together 1,576 human transcription factors. We think this is at least one per gene that is classified even in a broad classification of what a transcription factor is. 88 of them have alternative splice forms, and we're finding that those are quite useful, and we'll probably keep expanding that. But there's at least one transcription factor per transcription factor gene. And we've had many applications. We have even though it hasn't been published, we have already 8 collaborators that found out about it through talks like this. And here's an example where we screened it for loss of pluripotency in a human stem cell line. More about that in a moment. And what you can find is at one end of the spectrum are things that stabilize pluripotency, and at the other end of the spectrum are factors that take it to differentiate into something. And what it differentiates into is an interesting open question, but we've used this to find a whole variety of new pathways, some of which are extraordinarily efficient and rapid. So, for example, when we were some of the protocols we're looking at for some of the tissues the protocols in the literature would require 120 days and maybe have single-digit efficiencies. While here we have now numerous transcription factors that will give us 98% yield of neurons in 4 days. You'll see a time course in just a moment. And so essentially we don't even have to remove the stem cells. They're completely gone. And the 2% that aren't neurons are not stem cells either. But the point is it's very efficient in a very short period of time. And here's the two methods that I've alluded to that we've tried. We developed one of the most efficient CRISPR-based, so this is dCas9, meaning a dead Cas9, with 3 different activation domains attached to a C-terminus. And this is about 20,000 times more active, or can increase gene expression by 20,000 times. And it's 100 times more active than the first CRISPR activators that we and others developed. Anyway, when we hook this up to neurogenin, we get about 7% yield. But when we go to neurogenin directly from both the doxycycline inducible, but directly to transcription factor, we get 98% yield. And you can see that within a day, we're already seeing this huge transformation from sort of this gregarious pig pile of stem cells, to these elongated bipolar neurons, where you have exactly 2 processes. Now, one of the challenges when we develop these protocols, where we get a new developmental fate, is figuring out what it corresponds to, because unfortunately, despite all the transcriptomics that's occurred in the world, we still don't have a list of all the transcriptomes for all the cell types in all the stages of development. So we don't know exactly what this cell corresponds to yet, but we and others are working on a cell atlas for human and mouse that will cover hopefully every cell state in every stage of development. Most of the transcriptomics, unfortunately, has been done on blocks of tissue that have multiple cell types. Anyway, so that's the conversion. It's very fast and very efficient, and we've done it for a large number of cell types: neurons, myocytes, endothelium, oligodendrocytes, and so on. And here's some of the cases where on the top are the genes, the transcription factors that we've used, and down below are some of the antibodies that we use to show what cell type it is or to do FACS sorting. And then we follow up with transcriptome on these pure cell types. And this was work from Alex again and Parastu, who's a postdoc in the lab. Now, the next step up from cell types, tissue types, is organs, or organoids, as they're modestly called. So organoid means we haven't really gotten perfect organs yet. And there's pioneering work from Lancaster and Noblik in 2013, where they actually use these to study microcephaly genetic influence, essentially show cause and effect of microcephaly variation in a way that's very hard to do in mice. It's hard to get because of the anatomical structures are not present. But they actually got good anatomical structures. You can see here this beautiful layered effect. But if they get more than 0.5 millimeter, you start getting a necrosis because there's no vasculature. So we set out together with Mark Scott and Jennifer Lewis to solve this vasculature problem. Again, using the same kind of pluripotent stem cells, or PSCs, IPS, human IPS, that we were using before, but now using the transcription factors in multiple combinations to get at vasculature. Now the challenge here was if you just take neurons or neuronal precursors and endothelial cells, even immature ones, and mix them together and hope for the best, they will separate like oil and water. That's been our experience. But if you put in induced pluripotent stem cells that are undifferentiated, they love to aggregate, and if you have a mixture where they're programmed in advance to do different things when you add the doxycycline inducer, then you'll get this aggregation, they won't segregate out, and you'll get interspersed, self-assembling, now vascularized organoids. So I already said the Lancaster et al. Paper had organoids, but now you have this organization with vasculature mixed in, the capillary bed. And so here, the capillaries have been here labeled with antibodies to VE cadherin, and you can see they're nicely interspersed with the various neuronal types. We've also, in addition to the capillaries and the neurons, we also have oligodendrocytes and other non-neuronal components. The next step is hooking this up either to micro pumps, such as the ones that are already common in our lab and in Jennifer Lewis's, or getting it to pump itself with a cardiac muscle that you'll see in a subsequent slide. Now, just a slight digression here. All of the human epigenetics that we've shown so far is due to the Personal Genome Project cohort that Francis mentioned in his introduction, and all the slides I'll show for the rest of the talk are also from the same cohort. A good number of them are from PGP number one, guinea pig number one, which is me. And it's had advantages. To take two examples here, the NIH has an ENCODE project, which is Encyclopedia of DNA Elements, which is aimed at observing the epigenetics in every way you can, looking at factors, proteins that bind to different base pairs throughout the genome, looking at RNA levels and so forth. But the project was mostly predicated on whatever cell lines were convenient at the time and sort of represented various tissue types. But this is the only one that I know of where we had isogenic sets such as fibroblasts to stem cells to bipolar neurons, where these all had the same genome but different epigenomes. And so I think that made a particularly nice set, and it's publicly available, the data from all the labs that were involved in the NIH ENCODE project. Another project was a collaboration between NIST and FDA. They don't collaborate that often. I think this may be the first time. And they established Genome Model to establish standards for the world for providing genomes and cells and so forth. And they looked around the world for a cohort that had been properly consented for this sort of thing, and ours was the only one that had been consented for possible disclosure of their identity and also for commercial use, because these standards have to be used commercially. So anyway, this project is now international: Boston, Toronto, London, Vienna, Ulsan, and New York. And in the case of Austria, it is synonymous with the entire national genome project. It's called Genome Austria. And it's collaborating with many other omics. This is, and any of you are invited to join it. This is another example of use of these P2P cell lines. This is now for getting at variants of unknown significance. It illustrates, in a certain sense, you run it one way and you have gene therapy, you run it the other way and you get a cause and effect of individual point mutations. Bill Poo did the clinical genetics on this cardiomyopathy, which he thought might be a mutation in the TAS gene on the X chromosome of this little boy. He hypothesized it might be this guanine. Now, in some cases, the hypotheses are not this narrow. There's hundreds of potential candidate alleles, but we picked this as a nice case. So the X chromosome, there's only one copy of this gene and only one base pair. So this is one base pair in the whole 6 billion we thought might be causative, but up to that point, we didn't even have correlation data because this was an N of 1 study. And you say, well, what can you do with an N of 1 study? And the answer is, I think, quite a bit. On this particular case, we turned it into cause and effect by taking these guinea pig number 1 cell line, putting in the CRISPR, humanized Cas9, along with a repair oligo. If you don't include the repair oligo, you get this mess that some people call editing, but I call genome vandalism. But if you do include the repair oligo, then you get with fairly high yield, sort of in the 30 to 80% range, depending on the tissue type, but you'll get a deletion of the G. An example of this genome vandalism is an insertion of 8 random base pairs that have no match, anything obvious. Anyway, so you can make this 1G deletion. Now, and then you can make organoids. So you've done genetic change, and then you do epigenetic reprogramming, and that's what we have here, is along with Kit Parker, we've made cardiac muscle here, and you can see this beautiful repeating motif of the sarcomere organization of this cardiac muscle, which will contract normally with sort of a 60 beat per minute rate, and then one base pair in the whole genome, just changing that one base pair causes abnormal biochemistry, morphology, and physiological beating, as does inserting 8 base pairs. They're both out of frame for the TAS protein. Now you might say, well, you've talked to me about off-target and on-target problems with CRISPR. How do we know that this is really what you say it is? And the answer is that my graduate students think nothing of when they change one base pair of sequencing all 6 billion base pairs to make sure they did the right thing, because it is inexpensive now. And that might be a lesson that someday we'll think nothing of synthesizing all 6 billion base pairs just to change one base pair if it's cheap enough. Stranger things have happened. In any case, this is kind of proof in an N01 study that you have cause and effect of that single guanine for that cardiomyopathy. So back to brains for a moment here. We are not just interested in making brains. We also want to be able to make sure that when we make these organoids, they are physiologically reasonable, and if they're not, we want to debug them, and we want to debug them with all the tools we can, and we call this A through E activity, which is typically done with calcium imaging, but we're also developing methods where we can do it in situ, and time does not permit, but questions can ask. And then we monitor behavior, in a case often video input for visual tasks and video of what the motor responses are. Then the connectome, how the synapses are connected, the developmental lineage, how each neuron or each cell in the body got to where it was from the fertilized egg, and then finally expression analysis of RNA and protein. This is funded by IARPA in the Microns project as part of the BRAIN initiative, and these are some of my colleagues at Cold Spring Harbor in Columbia and MIT, and the main thing is getting this connectome and correlating it with the activity map with calcium. This and essentially all of the things on our A through E list are helped by the fact that next-gen sequencing is actually an imaging method. When we first developed it in 1999, we literally used an Icon microscope, which we souped up and automated, but it was essentially a microscope, and it still is. But conventional sequencing, you randomize the cell contents and splay them out randomly on a flat surface. The original concept, even before next-gen sequencing, now called conventional sequencing, and what we have now implemented called in situ sequencing, is to leave the cells alone, leave them on a surface. They can even be fairly thick, tens of microns thick, and sequence them where the RNAs and DNAs and proteins lie in the first place. But in both cases, it's just a series of cycles where you use 4 colors per cycle, and you build up, and you get barcodes that essentially have an information content of 4 to the n, where n is the number of cycles. And it's easy to do hundreds of cycles with next-gen sequencing, whether it's in situ or conventional. We tend to aim for 30 cycles or less because you can get all the information you need from a short tag. Now this, I'm going to show you multicellular in situ sequencing, and then later I'm going to show you subcellular because RNAs and proteins are not uniformly distributed, and cells are not spheres where you can just take them out and analyze single cells. You have to see them in context. And here you see there's 4 colors, A, C, G, T, and each neuron has a specific color because this is we've barcoded the entire neuron. When we get to subcellular, you'll see there'll be thousands of dots within the cell, but for this purpose, we're barcoding these neurons so we know how they connect over very long distances, sometimes centimeter distances. And this is work from Tony Zader's group. So now watch on the side, we're just monitoring neuron 1 and 2, and we're building up a barcode. And the cells, all the processes will stay in place, but the colors will change. And you can see all the axons and dendrites, and you can see the barcodes building up on the side. Now this is subcellular in situ sequencing. This is, each dot there is a single RNA molecule, and as you saw that the dots stayed in place, but the colors kept changing, and then we can record all those colors and then play them back out of the plane as a barcode. So every position in there had a barcode, which was built up the same way next-gen sequencing, except we didn't bother to throw away the morphology and the 3-dimensional coordinates of each of the RNAs. But it's not limited to RNA. We can also use DNA and protein, as you'll see in the next couple of slides. And we can do super resolution as well. So there's 2 main ways we do super resolution. One is called STORM, which is a physics and optics strategy where you query over and over the same fluorophore, and you get a better and better centroid for it. And so here's conventional microscopy, and here's exactly the same thing, the same magnification showing STORM. And it's getting sort of in the 5 to 20 nanometer resolution, and so it's on the order of nucleosomes. And this oligopaints is sweeping through the field. It's a work from Ting Wu's lab, also a professor at Harvard Medical School. So that's RNA and DNA. Now proteins seems a little harder, and it is, because we're highly limited by antibody resources. But if you have these purple proteins and gray antibodies, these Y-shaped molecules, tagged with a oligonucleotide, which can be coupled to a polymer. So this illustrates 2 things. One is how to tag proteins and turn it into a nucleic acid problem. And the other is how to get expansion microscopy, where you literally take the cell and expand it by, this polymer is polyacrylate, which is the active ingredient of disposable diapers, which, as you know, will absorb a lot of water. And this will expand to 100 times its size if you do 1 round of expansion, and 100 squared if you do 2 rounds of of expansion. And so, for example, here's a pre- and postsynaptic proteins that are normally not resolved, that are beautifully resolved, and help us determine where the synapses are and which direction they go. And this is work that we've done for a few years now with Ed Boyden's group, now integrated with expansion. It's called expansion sequencing and expansion FISH for ways to integrate it with other nucleic acids. This is a busy slide, but the take home here is we have 9 different ways, just like we have 9 editors, we have 9 different ways of detecting metabolites, where we can actually turn a steady state metabolite level into a protein level by stabilizing that protein. And you've already seen that we can detect proteins in situ. So we are pursuing this as a way of doing metabolomics in situ. And so in summary of that part, we have what we call omni-omics, which is pretty cool all by itself, but you add the word in situ to it, and now you can do genomes, transcriptomes, proteomes, metabolomes, et cetera, in situ, where you have all the advantages of microscopy and morphology. So the last thing that I think we all should do, and Marshall Nirenberg did, is to engage with society as a whole, think about where this is all going, and certainly many of the technologies I've brought up raise issues, and since the technologists see them first, we have a special responsibility of pointing out the downsides and the solutions to the downsides, and then the problems that the solutions create, and then the further tweaks, which makes people nervous, but we need to do it. So first thing, I'll just jump right to what worries everybody is genetically modified humans. There are already tens of thousands of genetically modified humans because there are 2,300 clinical research trials over the years for gene therapy. Now, you may not say that, oh, that's not a genetically modified human, but it most certainly is. One of them is approved, and the delivery vehicle for that is adeno-associated virus, which explains why so many people are excited about AAV, because it's approved, and it's fairly benign in every way, but it's really terribly small. It's terrible in that regard. It is, hopefully the price will drop. I mean, as with orphan drugs, you have almost the same cost of development as a regular drug, but a smaller market. And for many of these, genetic counseling is an alternative. Okay, so you're saying that's not really genetically modified humans. It's about germline therapy. There are already people that have received mitochondrial DNA therapy. Somehow that isn't always classified as germline therapy. I think it should be. There's a possibility of using spermatogonial stem cells, which is not always discussed. Typically, the discussion is about engineering embryos, which is problematic because of mosaicism, where different cells in the embryo have different types, and so you can't really determine whether your CRISPR or other gene therapy worked. But with spermatogonial stem cells, you can get close to 100% correct editing by the method that I described earlier for the cardiac study, by analyzing stem cell clones. And I think this will deal with infertility, but also if it's applied to recessive genes such as Tay-Sachs, which already are being treated by either abortion or IVF-PGD, which some would classify as as even more embryos at risk. You might have 25% at risk without IVF-PGD, and you might have 80% at risk with it. So the irony is that germline therapy might reduce embryo harm rather than increase it. So I think we need to be not too dismissive, and we have to think very carefully about this. Amping it up a little bit further in terms of alarm level, there is this issue of can we alter behavioral and cognitive traits? This is often dismissed, and I think dismissing is not a healthy thing for either side, because it's hard to see it varying with alleles in natural populations. But in synthetic situations, just like you will see big impacts in rare cases, you'll find big impacts, and also in synthetic cases in a moment. So, for example, there's effects on Alzheimer's, and there's some that affect intellectual disabilities. But then there's gene therapy trials that are based on somewhat abnormal, not found in human populations, but based on genes that are in the genome. And so, for example, there's some gene therapy trials in either preclinical animal trials or in human trials. And there are even some that significantly enhance performance in cognitive tasks. And here's an example of one of those cognitive tasks showing the black ones where we have a significant increase. Very specific cognitive tasks, maybe sometimes multiple ones. And the reason that we might not be able to dismiss it as multigenics and hence very complex, meaning so many genes and environmental components, is that even though there are maybe 10,000 single nucleotide polymorphisms that affect things like height, sort of the classic textbook example of a complex trait with genetic and environmental components, it's not all about the natural variation in the middle of the bell curve. It's about the ends of the bell curve. And at the ends are things that affect both ends. There are things that affect growth hormone and growth hormone receptor. And when you find those, they're very often not only informative about the pathways, but also they provide therapies. And here's an example where growth hormone therapies work even on non-growth hormone alleles. And here's a series of clinical indications where growth hormone is used as a drug. Even more alarming is people, so we've gone from genetically modified humans to germline to complex traits to now do-it-yourself gene therapy. And this is a real thing. People are doing this. It's been reported in 2 articles in Technology Review, among others. I can give their names because they're public. And they're do-it-yourself CRISPR kits for $150, assuming no prior equipment whatsoever. And these 2 did gene therapy by outsourcing it. One of them doesn't even have any biological training whatsoever. Now, these were tested in animals, but not exactly the same gene therapy. So, and it's an N-of-1 study, which in this case I don't think is a good thing. The N-of-1 that I mentioned earlier is fine. But these were tested in all these different animals. And they're trying to reverse aging, and it's not completely out to lunch to do that. There's a great deal of information known about aging in model organisms. And in fact, our group has taken 45 of these into gene therapies for veterinary preclinical trials, which could then lead to human. And in fact, the FDA approval process for veterinary is much faster. And so I think this is a more cautious approach, but we'll see. Another application which has caused some concern, 15 years ago, there was concern that transplanting pig organs into humans would, in addition to immune problems, would have endogenous retroviruses that would infect human cells. And it is known that they do infect human cells. And so especially immune-compromised hosts would not be great. So when we brought CRISPR to bear on this problem 15 years later, so it was about $1 billion investment, and then it dropped because of these endogenous retroviruses, or PERVs. I didn't come up with the acronym. These were, we knocked out all 62 at once, and it was not a heroic thing. I'd love to make you think it was heroic, but 14 days in the incubator and then a little bit of PCR was all it was. And then we've also knocked out most of these 45 genes that are involved in the immune complement clotting incompatibilities with human. And so now these are fetuses. We have, this is not published, we published the endogenous retrovirus, which is a record for CRISPR 62 at a time when 2 or 3 genes at once was challenging. We have positive ultrasounds, but I won't believe it until the piglets are healthy and are used to transplant into non-human primates, hopefully later this year. And a lot of this was led by Luhan Yang, who was a postdoc and is now leading this company called Regenesis. Another troubling, positively and negatively, each of these has very positive components as well, is this idea of gene drives. This could be a real boon to public health, especially in countries that can not afford all other medicines, and even vaccines are hard to distribute into remote areas or war zones. Here you can aim at the vectors, and the vectors spread the good package themselves. The package in this case might be anti-malarial, multiple anti-malarial peptides or antibodies that spread through the mosquito population at not the normal Mendelian rate of 50%, but which doesn't really change, but at 100%. And here is an example of a standard drive. And one of the things we did in this case is we got ahead of the game and talked about the safety issues and reversibility and keeping things local before we started any experiments, rather than announcing it like a gain-of-function flu after we're all done. And then we did the experiments in a variety of organisms, ranging from yeast to the malaria mosquitoes. And the idea is that CRISPR cuts, you have in an essential gene and transfers over a copy of the gene drive, which includes the whole CRISPR machinery as well as your payload, which will fight the malaria, make the mosquito resistant. And in the standard drive, it cuts and gets replaced and repaired by gene conversion, and now you have 2 copies, so all the offspring, 100%. And then this is the both calculated and observed rate of spread. It goes to fixation quite quickly. And we've come up with a DAISY drive. I'm not going to go through the details unless you want to in the questions, where CRISPR-1 cuts site 2, CRISPR-2 cuts site 3 or A, and then they all decay because they have a slight disadvantage in this model. But A goes close to fixation and could even go to fixation depending on how many stages of CRISPR and how you rig this. But it can control things in time and in space. This is, again, in this lovely journal called bioRxiv. So in wrapping up, I want to say 3 thank yous. One is to pged.org for the ELSI component of this in education. They do congressional briefings without lobbying, about quarterly. They reach out to underrepresented minority high schools and work with screenwriters, and then they have these online game tools like MapEd. And this is initiated by Ting Wu in 2006. And Jean-Tine Lunshoff is an ethicist that's embedded in our group and has been working with us ever since our first SEGS grant had an ELSI component in it. And we've published many papers together, about 20 or so. And then finally, I want to thank the NIH for funding our SEGS, our Center of Excellence for Genomic Science, in this case, our 3rd SEGS on 3 different topics. First one was next-gen sequencing, the next one was, that was in 2004, then one on zinc fingers, which we didn't deliver on the zinc fingers, but we did deliver CRISPR, so I figure they gave us, they were okay with that. And also on the first one, we didn't deliver the 1.7 million base pair bacterial genome that we promised. We delivered 5 human genomes and next-gen sequencing. So we're getting away with murder on these grants. But anyway, the next one is on organs. And then I mentioned the IARPA project and I didn't mention 2 transformative awards for molecular recording and using this in situ method on human samples. So, questions? Thank you.
So please go to the microphone to pose questions so that everybody watching video can hear. While they're thinking about it, George, let me ask, I was very struck with this effort to find expressible versions of all the transcription factors, and that you were seeing some pretty surprising things from transfection. And obviously one wants to go from single factors to mixing them up. Yeah. And if you were going to do 2 at a time, well, that's about 2 million experiments. Right. Is that feasible?
Right. So, so what we're doing with the single factors for the library is we will take a target, let's say, and do RNA sequencing on single cells, so we know what the levels of the transcription factors are. We take the top ones and we put those in as a mixture, so we're not doing a full combinatorial. And then we'll put in the whole library as the unprogrammed part in case we miss something or there's something synthetic that can help us out. That's one of our current plays. But we wanted to know what they all did individually so that we can put that into the model. Sure. Pairs, we could probably do the whole pairwise. Triples, I think, is getting out of range. And that's where you use this other method where you put in as many as you can guess at and then put in the whole library.
That makes sense. Over here.
Yeah, from a past experience in pediatric pathology, I can tell you that a sacrococcygeal teratoma can regenerate all of the organs, essentially all of the tissues. And because I've seen a few of them, I can tell you that it's always a question why you wouldn't just want to inject some stem cells into people, generate a teratoma. My understanding is you put stem cells to a critical mass, they'll automatically convert to a teratoma. Maybe I'm wrong. And you can just pick the cells out of there to do your engineering, rather than to go through all this induced pluripotent stem cell, etc. I've seen it. You have perfectly viable neural tissue. You have all the tissues, skin tissue. So is there any answer to that, or hasn't anybody thought of it, or is that just not a viable idea?
Yes, that's a great question. In fact, I started my work with Gail Martin, who worked in teratomas in the 80s, and so I did my postdoc in her lab in 1985. And she was one of the first practitioners of embryonic stem cell manipulations back before it was popular. The problem, what we're trying to do is we're trying to make organized organs, not just tissues. We want to know the rules, and we want to know what we can do. And we want, the closer they are to physiological organs, so the teratomas are very far from normal morphology. They will have local pieces that are quite interesting, but they don't have the right, and so that's our goal. And I showed you an example where we're vascularizing and putting in glial cells into the previously fairly successful cerebral organoids, which were instrumental in figuring out microcephaly. But a lot of this has to do with the basic research of figuring out how organs assemble.
Okay, thank you.
It's an unusually shy audience, so don't be shy, folks. There's a lot of issues here that we could talk about while they're thinking. You showed the data on what you're doing in terms of preparing pigs to be able to produce organs that would be acceptable to humans, both because you've knocked out the endogenous viruses and you've also maybe tried to make them less immunogenic, although one wonders just how many things you have to knock out in order to fully achieve that and not require immunosuppression. Obviously, the alternative, which other people are pursuing, is the idea of making chimeric embryos where you end up with a purely human organ in a pig host, because the pig is engineered so that it cannot make its own heart because of the genetic defect. Paper in Nature describing this just about 10 days ago. So what's your view here, the pros and cons of those approaches, both of which, by the way, have some ethical issues that are quite interesting and vexing?
Right. Yeah, so I would add a third approach, which is making human organs entirely outside of an animal. But let's talk about those too. The problem with the chimeras is that, well, first of all, I think it's ethically more problematic because you, but in addition to that, when you take out one organ, you remove it from the developmental potential, and there's essentially a gap that is filled by the human stem cells. You really typically gotten rid of one cell type or a very small number of cell types, and there are many other cell types that are still in there: nerves, capillaries, various muscle types, smooth muscle, etc. And if you don't replace all of them, then you're going to have a host rejection of that part, and that's going to result in necrosis probably. So, and most of these have been over, the most successful ones have been over very short distances, like rat to mouse kind of distances. I'm not saying it's impossible, I'm just saying that these are some of the limitations. So if you're going to humanize them anyway so that they can take on, you can engineer them to take on human chimeric cells, you might as well humanize the whole thing. Because and the third problem is that if you if you've engineered one organ, say the heart, the rest of the pig is useless. But if you humanize the entire pig, then you can get 10 different, sometimes more, transplants. All the same tissues you would get from a human, you can get from a pig.
Over here.
Hello. The first is a very small question. You showed.
Closer to the microphone so we can hear
Yes, you showed at the beginning the abundance of the codons in the genome. Mm-hmm. Could you talk about how that relates to the abundance of the different tRNAs?
Right.
And also that relative abundance in different cells or tissues.
Right.
Cells, actually.
So, so this codon table is for E. coli, and there aren't really tissues, but there are cell types. And it does vary from, say, stationary phase to rapidly replicating. For the most part, the number of tRNAs corresponds to the abundance in this table. The exception is methionine, which is special because they're initiator methionines and so forth, but there is a correlation. And so when we recode, if we want to change it from one synonym to another, then we need to increase the number of tRNAs or release factors to compensate for that, probably. But they're definitely used for regulation, but it's kind of it's a nuance that we can layer on top of it.
Okay. Yep. And in different, sorry, and I also have a second question. In different cells, you find the same expression of the different tRNAs?
No, the tRNAs are regulated. Their levels are regulated depending on the cell state and the cell type.
Okay, and the second question is broader. How do you see all these genomic things converting into personal medicine? So in terms of public versus private research and how much these things cost and how it's going to develop?
Yeah, that's a pretty big question, but I'd love to talk to you or a bunch of you afterwards. But I think that we have a huge opportunity here with the technologies still going exponential. I think we're going to very soon have, in addition to two cameras on your phone, you're going to have a sequencer on your phone too. The nanopore technology that I didn't talk about is really going in that direction of portability and low cost. And so you can analyze your environment, which has been something that's been hugely missing from the association studies so far.
Over here. We'll take two more questions.
Hi, Dr. Church. Thank you for the talk, first of all. I have a question regarding building the organoids. So one of the progress you were talking about was building vasculature into the organisms you were growing and how that was a challenge. My question is, do you think building the entire, say, a fully functional brain, each of the sort of units have a recursive programming to it, meaning that they self-grow and they sort of, in the end, organize into this beautiful structure of the brain? Or do you think that somehow at some point there's a directive or a plan that sort of shapes the final product? So whether this is more individual blocks forming together into complexity or more a master plan directing?
Right. So another big question, but it's an open one, and it's even more open than it might seem because we have the way it normally happens and then what synthetic biology can do with it. And we've been pleasantly surprised numerous times now with our synthetic developmental biology that we can take things that normally take hundreds of days in vivo and even hundreds of days in most protocols and shorten them to 4 days. In fact, 4 days is kind of our standard for many of these protocols. We need to be vigilant that what we're getting is either natural or acceptable. And in particular, making a human organ, typically an adult-sized human organ, takes on the order of decades. Are there shortcuts, or are we going to have to go? I mean, clearly the pig does it in less time than we do, and makes something that's quite large. So it's a really interesting question: is can we use synthetic approaches to making organs faster and possibly better? And by better, I mean, reduced cancer, reduced viral, and reduced senescence. That's something that would be very hard to do in humans, but so the pressure, essentially the approval pressures are against it, that kind of preventative medicine in humans, but they would be in favor of it when you're doing organs. So I think there's some really interesting opportunities.
Thank you.
Well, you covered the world of genomics and also ultimately the human frontiers of brain. So you think someday we should be able to identify clusters of genome for higher intellectual ability of cognitive processing and all the other neuronal functions?
Well, there's two shoulds here. One is technically and the other is societally. My guess is that both will happen. And part of it is there's going to be high motivation to deal with Alzheimer's and other neurodegenerative diseases. And some of those solutions will be effective in people who are not already degenerating, and they will actually be enhancing. In the same sense that vaccines are enhancing, they make us really super immune to a variety of pathogens. And we've already seen in mice that if you have a specific set of cognitive tasks in mind, you can, with 1 or 2 genes, make a huge change. So I don't think it's going to be fundamentally different from other things. And we shouldn't use natural variation in the population as our only guidepost, because there are synthetic things that you can do. In fact, most of pharmacology is not natural. And you end up with things that are, in some cases, far beyond the native state of human beings. But we need to think about this very carefully and talk about it in advance, as we're doing today.
Congratulations. Thank you.
So what a wonderful romp we've had through technology, visions of the future, ethics that has to be very much incorporated into all of this, which George has emphasized throughout all of these adventures. I think if Marshall Nirenberg had been here, he would have loved this. So please, we can join now in the library for some coffee and cookies, but meanwhile, please, let's thank George Church again.